Everyone. So I have the pleasure now of introducing Marty Jobson for his second session in STEMtastic. He did one on Monday called ZAP, all about high voltage electricity and things, which was really interesting. Marty Jobson is he's done science television stuff for over 20 years. He's been a presenter in different things for over 10 years. He's the science reporter on the One Show. He's done things like Invention Nation, The House That the 50s Built, Food Factory and Brainiac. He's done so much stuff. It's amazing. I'm very jealous of the career that he's had so far. And this one is all about invisible worlds. So this is all about those little things that you can just about see, I believe. So with no more from me, I will pass you straight over to Marty because I know he has lots of stuff to try and fit into his slot. So over to you, Marty.
Brilliant, thank you very much. So, hello everybody. I'm just checking all my buttons and my dials and making sure something collapsed just at the very last second. So, always a technical glitch. Right, so yes, my name's Marty Jobson. I'm a science communicator. I do a lot of weird things on telly, but I originally trained as a plant cell biologist. And actually, I trained using machines like this. These are microscopes, and I've subsequently returned to, as it were, my first scientific love, microscopy, and now I spend a lot of time talking about microscopes to people sort of out there on the interwebs and in person as well when we ever get to do that. Now, if you've got any questions, put them in the Q&A. I've got the Q&A open on my computer here, and if I put my glasses on, I will actually be able to see what you put up. So any problems, just stick them in there, and I'll try and answer them as we go along, and if you've got time at the end. So I'm going to take you on a bit of a journey. I'm going to take you on a journey from small stuff to very small stuff to ridiculously small stuff. And we'll sort of hopefully see everything in between. And I'm going to be using two different microscopes. This one here, this one's really good at looking at actually quite big things, sort of a few millimeters. And it's really good because you can put the whole object underneath it. You can sort of stick a whole animal underneath it there. Whereas this one, sort of microscope you're more used to seeing, this is called a transmission or a compound microscope. And it's for the very small stuff. So let's start with a very simple sample. This is a sample that I fished out of my garden pond this very morning. I went and I had a look at the garden pond and I pulled out some weed. Now, it's actually just been raining a lot. I'm up in Yorkshire, which is not good for what I'm about to do, because what it does is it knocks all the interesting critters down in the water in the pond. But I managed to find a whole load of pondweed. If I pop that under the microscope, I can show you. I'll flip to the microscope like this. Here is the microscope. Oops. Hold on, I'll do that. Then I do that. And I have to press that, sorry, too many buttons. So there you go, a little bit of pondweed. You've got sort of things you'd expect to see in there. You've got, this is duckweed, just little individual leaves. They're just starting to regrow. And you've got some filamentous algae there, if I turn that down a little bit. And you can see, if you watch closely and I sit still, every now and then something small kind of zips across the screen. Now, the pond water is full of creatures that I could show you, but I've chosen to show you just one. And what I did was, just before we started, I fished out a couple of specimens. So I'm going to get rid of that, and I'm going to introduce you to the Hydra. Now, for those of you who remember your Greek mythology, the Hydra is a Greek monster whose head you couldn't cut off. Here it is. So it's a lovely little thing. Its proper scientific name is Hydra viridissima. Viridissima obviously meaning it's green. We'll come to that later. And this thing is, it's related to a jellyfish and sea anemones. And okay, here's the thing about microscopes. Microscopes are, and I'm just going to adjust one of my settings. Yeah, microscopes. Often people think about what is the magnification you're using? How much magnification? People worry about magnification. But actually, half of what you need in a microscope is actually illumination, because if you can't illuminate your specimen properly, you're not going to see it. Now, what I'm going to do is I'm going to illuminate this specimen using a special technique called dark field illumination. At the moment, the light is coming basically straight up through the specimen. But if I change that, I'll just zoom back out a little bit. And yes, I know I'm not supposed to use the word, but some biology teachers get very upset if I say zoom. It's what I say, okay? Live with it. So I've changed the magnification. If I change it to dark field, rather than the light going straight up, the light comes in at a funny angle, a dark angle, and suddenly, pa-ching, it pops out at you, and it becomes so much more visible. Now, what I wanted to show you though, is this thing, it's quite responsive. I've got here just a little sort of pokey stick. And if I just give it a gentle poke, watch what happens. Just give you a little tickle, there you go. It kind of pulls itself back in. Sometimes if you really hassle it, it pulls right back in like that. There you go. And it's fixed. It's basically a little tube of jelly. And do you notice how it's immediately started to stretch back out again because it's hunting for other little animals? And you know that it's actually, it's gone in the other direction. So it was kind of coming out, which way? Sort of, oh, it's very difficult because everything's back to front. It was sort of coming out in this direction here, okay? And now it's switched and it's going in that direction, okay? It knew that there was something hassling it from that side, so it's moving in the other direction. It's moving away from the source of irritation. So this thing has the ability to make decisions. It's also an astonishing creature. It's one of the few creatures that displays what's known as totipotency, which means that basically you can take a cell from anywhere in its body and that cell can be used to grow another one. Now, some people will tell you that, you know, if you take an earthworm and you cut it in half, you get 2 earthworms. That's a lie. If you take an earthworm and you cut it in half, you have 2 dead halves of an earthworm. If you take one of these critters, Hydra, and you cut it in half, it will regrow. The top will regrow a bottom and the bottom will regrow a top. Not only that, it's immortal. They don't age. Just think about that for a second. If it's not squished or eaten or dried out or completely destroyed somehow, if you just have one in a pot with food, and it will just keep growing. And it doesn't seem to ever age and die of old age, which is quite weird because most animals do. It has an astonishing life cycle. Basically what it does is it catches prey with those long tentacles, shoves them in its mouth in the middle, digests them, and then spits out the bits it doesn't want. And I wanted to show you what it looks like under a higher powered microscope. So what I did was I took one earlier and I mounted it on a glass slide. This is a special, I'll show you what it looks like, just to prove that it's actually there. It's in a bit of a huff with me because, you know, I've just been, but there's another one, okay? It's just a little baby one. It's actually quite small. And it's mounted on a glass slide that's got a little well in it like this with a piece of a cover slip over the top. So there's only a tiny little gap that it's in, but that allows me to put it on this microscope. And it's starting to do its thing. There it is. So let's pop back to this picture here so that you can see what I'm doing. And I'll scooch over and I'm going to try and find it. This is the difficult bit on this microscope. There it is. Yep, there it is. Okay, so let's jump to this microscope now. So this microscope here, ooh, I need to do that. And there it is. I'd say she, but I don't know. And there it is. You can just about see it's green. Not so much light coming through now. But if I change the way we illuminate it, and again, it's all down to the illumination. If I use what's known as phase contrast illumination, we can see more detail. And I'm going to zoom in on it. And I'm going to enhance it. Right, look at that. Oh, by the way, oh, what's that? That wiggler, that is a dinoflagellate. There you go. Who knew? Okay, let's go and look at the tips of one of these tentacles, because the tentacles are armed with the most astonishing array of deadly harpoons. Okay, and if you look very closely, especially at the ends of the tentacles, can you see all these little sort of blobby cells here? Okay, so it's full of cells, and at the end there are blobby cells, and those blobby cells are these harpoons, which if it touches anything, if one of those little critters runs into it, it will trigger what's known as there is a trigger hair. Oops, where are you going? Come here, come here. A trigger hair, and you can see them. Can you see? There are a few little hairs sticking out there. If it triggers one of those, what will happen is a harpoon will literally shoot out. And sometimes you can find the harpoons resting on the bottom of the slide. Can I find one for us? I'm going to have a little, should we see it? No, I want to jump to one of the things. Sometimes I can find them. I've lost it now because he's gone away. Come on, adjust the brightness. Where are you? Where's he gone? Where's he gone? Oh, there it is. There it is. There it is. It's stretched out nicely. There it is. Let's zoom back in on that, those tentacles there. And he's playing hard to get now. This is always a hazard when you're doing, and this is, I'm doing this for real. This is not sort of pre-record. You're seeing this for real. And he's gone. It's upset. I've upset it with all the bright lights. But what the other thing I wanted to show you is another thing that I can do with this this microscope is I can use a different kind of light to shine on it. And what I'm going to do is I'm going to shine ultraviolet light on it. Okay, ultraviolet light, invisible lights, it's the stuff that causes, you know, skin cancer and sun tanning and stuff like that. But if you shine ultraviolet light, okay, onto some things, they glow in the dark, don't they? You get sort of glow in the dark stuff. Now, you noted that it was green. The reason it's green is because this organism has it's what's a symbiotic relationship with a kind of algae, okay? A single-celled algae, which is a green type of, you know, it's a primitive type plant like seaweed, but a single-celled type plant that grows inside it. And if I turn on the ultraviolet light and adjust the filters, all the chlorophyll inside that single-celled algae, and it's just white balanced itself, so we've lost the red, but it's glowing pink still. All of that chlorophyll shines up, and every single one of those little specks, right, is a packet of algae growing inside the Hydra, okay? And you can see all of this using, when you've got a decent microscope like this, using all the different clever ways of illuminating stuff. And this is where we are at the moment with microscopy in sort of research science. People use fluorescence microscopy all the time. It's a super common technique. And all I need to do is turn it off and go back to the ordinary light, is do that, and pow, we go back to that, and there he is. There is our Hydra flailing its tentacles around. Rather fun little specimen. Right, let's jump out of that and come back to me here, because, okay, people asking, is it like a polyp, like coral? Yes, related to coral, coral polyps as well, all in the same sort of little squidgy gobbets of jelly, kind of part of the animal kingdom. Sea anemones, jellyfish, stuff like that. And they're super common in your pond. And obviously it's a millimetre long. It's not going to do you any harm. People are often like, oh, is it going to harm me? Those harpoons that shoots out deadly neurotoxins. I've seen it work. I've seen it actually catch a little creepy crawly that kind of buzzed into it and went poof, and it was dead instantly. Okay, but obviously you and I, much too big. Let's talk about something else. Let's move on to something smaller than that. Pretty tiny. Let's move on to something much smaller. What I wanted to do is I want to show you some human specimens. And the obvious thing to look at, because it's really easy to get hold of, is human blood. So for that, what we're going to do is I'm going to take a little blood sample. Right, let me move my pots of pond water out of the way. And let's start with, get a couple of these. I'm going to need, go to the desk camera. So what we're going to do is I am going to take a sample of blood, my blood, obviously, because no one else here, everybody else is out. I'd love to be able to use somebody else's blood. It's got to be mine. This thing, it's got a spring-loaded catch at the front there. Okay, and this is the sort of thing that people who have diabetes use when they want to do a blood test. And basically it shoots a needle out of the little hole at that end there. And you can change sort of how deep the needle goes into you. I've got it set to maximum depth because I actually need a reasonable amount of blood. So let's do this. Okay, I don't like doing this because it generally is quite painful. There you go. Nice little spot of blood. There you go. Yeah, blood. Gross. I've just bled for you. I hope you appreciate that. You don't see that very often in a talk. Okay, I'm going to put the blood onto this piece of glass here, like that. Put it on there like that. just a little drop, don't need more than that. And then what I'm going to do is I'm going to try and stop myself from bleeding by sort of just pinching onto there. And what we need to do now is smear that blood out. We need to, because at the moment it's a huge pile of cells, blood cells, tiny, tiny things. It's a massive, great lump of cells there, millions of cells probably there. The way you do that is you take a second piece of glass and you do that. Simple as that. And what I've done now is I've smeared out the blood, and hopefully at this end here, you can see it's quite thick here still, but right at this end here, it should be just a single cell thick. And I've got to just talk for a little while once this dries out a bit. You don't need to put it under a cover slip or anything like that. You just dry it out. And that's dry enough now. I'm going to pop it under here. Let's go back to this picture here. Okay. And I'm going to have a little look, because this microscope we can all see simultaneously. Right, okay, good, I've stopped bleeding. This microscope, I have a look. When I found what I want to show you, then we can both have a look, because if I don't, what tends to happen is everyone gets a bit sort of seasick. It's quite unpleasant, actually. Right, okay, I've definitely got some blood. It's not the best blood. If there's any sort of hospital technicians in the house, then they will be like, didn't do a good job of that. And no, you're right, I did not. It's a bit thick, but we'll have a look at this. Okay, so this is all a bit sort of thick at the moment, but that's all right, we can live with that. Okay, there you go. Oh, that's good, this is good, this is good. Right, kapow, 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 kapow. Oops, and I want to be on that one and then do that. And there you go, there is my blood. Or at least my red blood cells. There's a few things you might be thinking. You might be thinking, well, they're not very red, are they? And you're right, they're not red. And in fact, red blood cells, when you look at one of them, sort of under a microscope, it's not red. I mean, it's ever so slightly pink. This camera here, it's very clever. It sort of auto white balances and does all sorts of clever things like that, unless I go in and change it, which is very time consuming. I don't have time to do that. So when you look down here, it is ever so slightly red, but when you look with the camera, you lose that red. And but the point is, one blood cell on its own is not going to be very red. Now, the redness, you might know, is from hemoglobin, which is a protein that contains an iron atom in the middle of it. And that protein, that heme group, is red. Okay, it's more red when it's got oxygen bound to it, which is obviously the job of red blood cells, is to move oxygen around. But there's actually not very much hemoglobin inside one red blood cell, so it doesn't appear very red. But if you pile a million of them up on top of each other in a drop of red blood, now you can see the redness. So to answer Margaret's question, why are they not red? They are red, it's just they're not very red. And when you look at blood, you're looking at millions of red blood cells, so you see more redness, so it appears more red. What you normally see when you look at red blood cells, though, is actually not this picture. What you normally see is a completely different type of red blood cell preparation. And I'm going to show you what that is, although I do have to put some gloves on for this bit. So let's go back to the desk camera. Will that work? I have to do that and then that. Oh, no, I don't do that. Sorry, I've turned that off. Okay, and what we're going to do is I'm going to stain the sample. Okay, and this is what normally happens when you look at red blood cells. So that's just literally dried red blood cells. I'll put these gloves on. And the reason I'm putting these gloves on is because what I'm going to do now is I'm going to take my sample. Here it is. Okay, my blood, and I'm going to dunk it in these three chemicals. And the way I like to describe it is this sort of like painting by numbers, because what we're going to do is I'm going to put it in one of each of these three pots, and then we're going to rinse off the excess and blot it off onto here. And what that will do is that will colour in different parts of the cell, because this is a stain that we're using. So the first chemical is this one here. This one is sort of purpley chemical. I'm actually running out of this one, so I have to go in at an angle. And I need to do it for the count of 5. 1, 2, 3, 4, 5. And then we'll just dry that one off. Just let that one run off. Then we go into this one here. Now, this pink chemical is called Eosin. 1, 2, 3, 4, 5. And Eosin, okay, Eosin is a chemical that sticks to the cytoplasm. So inside every cell, there's all sorts of smaller bits. And there, the pink stuff sticks to the cytoplasm, which is the sort of the general liquidy goo inside a cell. Now into a blue chemical. 1, 2, 3, 4, 5. And this chemical is, well, it's similar to, it's not exactly, but it's very similar to something called toluidine blue, which you might encounter if you go on and do biology A level, for example. And now I need to wash off all the excess because I've got way too much on now. I need to clean it all off a little bit, which I'm doing, like so. And you can see that and now I just have to make sure I wipe the right side. I do need to dry this off a little bit. I'm trying to show you what I'm doing here. Right, I'm just going to try and blow some of the water off. This might be a horrible loud noise for the microphone if I cover the microphone. Right, and then again, we'll try and dry that out there. Can you see that? I'm trying to see if it's drying. Yeah, it's drying slowly. So we'll dry that out a little bit. Another blow. Okay, that's better. Right, now what's happening, and you can see it's really stained up. Now, the toluidine blue, the blue chemical, stains nucleic acids. So that should stain DNA, okay? So that should show up the cell nucleus because that is the organelle inside the cell that where you'll find the DNA. So let me go back to this and I'm going to have a look and see if I can find what I need to find. Again, excellent, first time, brilliant. Let's see, we got what we need to see. Yeah, we have. Oh, actually, this is a good bit here. Right, okay, cool. I'll do that, and then I'll do that, and then I'll do that. And again, my camera is trying to, and you can see now everything looks a bit pinker, doesn't it? And that's because of the pink chemical. The eosin stains the inside of the cells pink. Okay, so we've got pink cells, and this is when you see something on CSI programs or something like that. Take my gloves off. What you're actually seeing when you see those sort of shots is you're seeing cells that have been stained pink with eosin, because I showed you what red blood cells look like when you didn't stain them. Now, you can see something that's showing up purple as well, because remember, blue and red will turn purple, and purple stuff is actually again color balanced a bit weird. You see here. And here, there's only 2 blobs of this stuff. This is the blue stain staining the cell nucleus. And you're thinking, well, hang on. First of all, why aren't the red blood cells staining up with a cell nucleus? Red blood cells don't have a cell nucleus. It gets pooped out just before the blood cell is released into the blood, okay? So when they form in your long bones, in your leg bones, they have a nucleus. And then just as they're about to sort of go off and become a blood cell, they've graduated from blood cell university, the nucleus is pooped out, which is why the blood cells have their characteristic kind of collapsed football shape, where it's rather than being a nice sphere, it's a kind of a squashed donut sort of thing, because they've gotten rid of this cell nucleus. The 2 cells that you can see that have got a cell nucleus, though, are, number 1, a lymphocyte, okay? Possibly, so one of the cells that's part of your immune system that's responsible for making antibodies, okay, and coordinating the immune response you have to an invading pathogen, okay? And this one, that one is a neutrophil, which is a type of white blood cell, okay? So it's another type of white blood cell. There are loads of different types of white blood cell. It's really complicated. The immune system is fantastically complicated. And these ones, this one at the bottom, the neutrophil, is responsible for hoovering up things that have been identified as being foreign bodies. So if there's a virus-infected cell, for example, the antibodies that are made by this bit of the immune system will stick to it, and then the neutrophils will come along and break up that cell and destroy it. And if they find any bacteria, they will eat those up. Anything that's not supposed to be in your blood, or in fact in any of your tissues, because these things go from the blood, they will actually go out through the walls of the blood vessel and into the tissue, hunting out things that are not supposed to be there. So I've answered that. Yes, will I show you to a microscope? The McNeil family say, yes, I did. What are the smaller circles next to the blood cells? There are a few little bits here. Can you see these? There's a few little dotties here. These might be platelets. It's hard to know without doing a better stain. Okay, and there's sort of some here. There's a few sort of clustered here, which are clearly not red blood cells. Those might be platelets, and platelets are what help blood clot. Okay, they coordinate clotting of blood. So when you get a scab, basically, when the blood oozes out and then it clots, that is caused by platelets. That's anonymous attendee asking that. Okay, so I've shown you some of my blood. Blood, I've bled for you. I'll put that down if anyone wants to come back to that at the end. But those are pretty big. Red blood cells are 8 micrometers across. So a micrometer is 1,000th of a meter. Millimeter is 1,000th of a meter. And a micrometer is 1,000th of a millimeter. Okay? And a red blood cell is a really good one, is 8,000ths of a millimeter. If we wanted to see something smaller than that, like, for example, bacteria, we need, first of all, to find a sample of bacteria. And secondly, what we need to do is we need to use yet another type of technique, because bacteria are super hard to see because they are a micrometer across. So if you thought red blood cells were cell, these things are far smaller. So what I've done is I've taken there just a glass slide, put a bit of water on it, and I've got here a cotton bud. I'm going to just wet that completely, and then I'm going to excavate out some cells from the inside of my mouth, and hopefully they will have some bacteria on the surface. Simple as that, doesn't hurt. Just extract a little sample, and then we're going to squidge that about on there like that. Okay, and hopefully some of those cells will now be on that glass slide, and there's just a tiny drop more water there. Okay, and I'll put that there. And then again, we put a thin piece of glass over the top called a cover slip. And normally you do this carefully. I don't have time to be careful. So I'm just going to whack it on like that. And then you blot the extra liquid away like that, so that now I should have some cells in here. And we're going to pop it under the microscope, and I will have a look and see what I can see. Now, the technique we use for this is a technique called, oh yes, lots of cells. As always, I gave generously. Have we got what I'm looking for? Yeah, we have. Ooh, no. Where are we? There. Yeah, good. Close that down. Yes. So what I'm going to do is I'm going to find a clump of cells, and I'm then going to zoom in on those cells, and I'm going to use this special technique called phase contrast. And phase contrast allows us to see things that you wouldn't normally be able to see. Bacteria are so small, they basically don't get in the way of the light. And what I'm doing is I'm just trying to find some bacteria, because I forgot I was doing this morning. I got up, and normally when I'm doing this, I actually go out of my way not to brush my teeth, to make sure that there's lots of vile bacteria all over the inside of my mouth. Okay, there's a few. It's not great. Yeah, let's see if we can find some more. No, I haven't got time. Let's do this. Let's go with these ones. So what I'm going to show you now, slightly confusing image, but we'll go with it. Right, here we go. So this is what my cheek cells look like. Okay, you've got, and these are living cells, by the way. They're not dead. They're completely living. You've got a cell here, and in the middle is a cell nucleus. And what I can do is I can focus up and curses. That, by the way, is an air bubble. But I can focus up and down on the surface of the cells. And if I'm very, very careful, I can find you some bacteria. So we've got a little clump of cells. So here's a clump of cells sort of scraped from the inside of my cheek. This thing here is an air bubble that I've got trapped underneath the glass slide. And if you look inside the cells, you see the cell nucleus and a lot of other sort of bits and bats. But if you look at the very top of the cells, so the surface of the cells, which is really hard to do. Can you see here, right, at the surface of the cells, there are lots of little black dots, okay? I'm focusing really carefully. And when I'm focusing carefully, I'm moving the focus knob less than 0.5 millimeter up and down. Okay, so this is really delicate work. Can you see here, there's a whole load of little black dotties, okay? Those are bacteria, okay? They are super small. They are really difficult to see. There was some others. There's a little, see right there, right in the middle where my sort of cursor is. Can you see there's like a little clump of 5 tiny little blobs? Those are Streptococcus mutans, which is the smallest thing that I'm likely to be able to show you. Those are the bacteria that cause tooth decay. So, okay, I've hit my half hour. I better stop. We've got a few extra minutes that we can do. Maybe we can do a few Q&A questions. But basically, I've run out of time there. So let me jump back to this. So I've taken you, hopefully, from my pond water. Where's my pond water? There's my little pot of pond water. And we had a look at the Hydra with its crazy tentacles and its poison and its sort of its ability to survive forever, its immortality, the immortal Hydra. We looked at it under ultraviolet light. We've looked at my cells, we've looked at my blood, we've looked at bacteria, we've looked at cheek cells. I've shown you the cell nucleus, all of which is very, very, very small. So Joe was asking me, and I'm just looking at the questions now. I'm just gonna plow on until I get hauled off by the organisers. But Joe asking, what were the cells with nucleuses, nuclei technically, that look like red blood cells? Those, well, some of the red blood cells had a tiny speck of purple in them, and that's because I didn't wash the slide very well. When you do these sort of things, you know, you should normally take half an hour, 10 minutes. You don't do it in 5 seconds flat whilst talking and trying to deliver at the same time. You concentrate and you do a good job of it. And then you get a really nice specimen with kind of, you know, each blood cell is a nice round blob and it's all just, and I didn't do that because I was hurrying. It's all a bit of a mess, but so some of them. What do red blood cells look like under UV light? Nothing. They don't show up at all, okay? Because there's nothing in red blood cells that will fluoresce. Okay, so when you shine ultraviolet light on things, it fluoresces. That means it kind of it shines back at you, basically. And there's nothing in red blood cells that do that, or even my blood, my cells here. So I could do it, but it would just be black. You'd see nothing at all. What are the small things moving? Ugh. Yeah, there are small little grommets moving in there. There might be bacteria, but actually more likely, whenever you put something under on a slide, what happens is there's always a bit, the liquid takes a little while before it stops moving around underneath the layers of the sandwich you made of glass. There's always a bit of flow of liquid from one side to the other. And when that happens, you get all the little bits of the lunch that I just ate that's still kind of knocking around in my mouth, the little bits of detritus flowing. So you see things moving. You can tell if that's what it is because everything moves in the same direction. If you see things kind of going and moving in opposite directions, you know that they are living and that they are actively moving, so to speak, in opposite directions. Yes, there's people asking technical questions about what to do. And somebody right at the beginning, anonymous, asked me, how much do microscopes cost? Right, these microscopes cost a lot of money. On this table right now, just the optics, none of the technology, because obviously there's computers and things, just these microscopes, not the cameras alone, you're looking at about £35,000 worth of kit. Okay, that's quite, I don't like thinking about it because it scares me to think that I've got these, which is why actually I'm going to pop this up now. If anybody wants more microscopy, go to my YouTube channel, follow me on Twitter. The nice guys at Zeiss who make these microscopes give me these because I couldn't afford to pay for them, just so that I can do this sort of thing. They say, here, Marty, take these, go show people lovely microscopy and make stuff, small things big for people. So have a look at those. You can just use I always recommend if people want to mess around with microscopes, the best thing to do is, number one, get one of those things that clips onto an iPhone, okay, or a phone. Okay, you just get a little, it's like a little peg, like a little clothes peg, and it just It's got a lens on it, and you do that, and then boop, you can see what's going on, and that works really well. I was just looking here. That works really well. The other thing is, if you want to go to the next stage, once you've played around with that, get a USB microscope. Because the thing about microscopes is the cheaper your microscope, if it's got kind of lenses and things, the harder it is to use. These things are a dream. They're so easy to use. You put something in, boop, there you go, image, beautiful images. If you've got a really cheap microscope that costs like £25 from Argos, you will struggle to see anything with it. It's really hard work. So just don't bother with those. Get yourself a USB microscope. They kind of look like sort of half a banana or something like that, sort of about yay big, and they've got a cable coming out the end. You stick it into a computer. You can use pretty much any software that looks at web cameras to see it, and it basically works like a webcam. It's got built-in lighting, it's got focus. You just kind of go dunk, focus, and there you go. You've got quite good microscopy. So that's my advice there. I'm looking, have I answered everything? Yeah, I've done that. Why they're not red? So, done that. What are the small things moving? Can we start recording? And then people asking about seeing the recording from the beginning, but I'll have to pass back to the organisers of and there she is, Catherine.
Yes.
Recording.
Thank you, Marty. I've been paying attention. I've been replying to a couple of emails, but I've been watching it all the whole time as well. It's been really interesting. I'm definitely going to have a bit more of a nose in. I've got one of those little USB microscopes somewhere, so I'm going to have a look at what's in my pond. Have a bit more look in there.
And yeah, have a go with that. They work really well.
Yeah. Definitely interesting. The question just about, can we start the recording from the beginning? Basically, this was live, as Marty explained right at the beginning, but it is being recorded and will be available on our website and things. So you will be able to see this again, and you can yes, watch it again. So thank you, Marty. That's been absolutely brilliant. It has been really good. So thank you, everyone. And I know there is another session coming up shortly as well. So hopefully everyone will tune into that one I'm not sure what the next one is, I'll be honest, but there is definitely another one coming up from STEMtastic. So thank you, Marty.
Take care, everyone.